All right, so to follow up from last class, we talked about DNA replication. In today's class, we'll actually be talking about transcription. On Thursday, the focus of the lecture will actually be on transcriptional regulation, and we'll be applying each of those to different contexts. Today, we'll be talking about transcription in the context of diabetes. Sorry, I started on the wrong slide. Bear with me. There we go. So, diabetes mellitus, what it is, it's a group of metabolic diseases, a group of forms of diabetes that are commonly characterized by hyperglycemia, high or elevated blood sugar concentration. And it all stems from either a defect in how insulin is secreted, the ability to respond appropriately to insulin, or it can be both. What's important to note is that typically when we consume food or drink that is rich in glucose, what ends up happening is that it'll get absorbed into our bloodstream, and from there, once glucose is detected, it'll prompt certain cells within our pancreas, beta cells, to secrete insulin. What's important to note is that insulin, one of its key functions, is to bind to insulin receptors on our cells and to enable glucose transporters on our cell surfaces to then transport glucose into the cell. And so that's once glucose is transported into the cell via these glucose transporters, that's when glucose can then be used as a metabolic substrate to then produce our key energetic our key molecule for cellular fuel, which is ATP. What's important to note is that in diabetes, excuse me, again, it's characterized by this chronic hyperglycemia, this consistent elevated levels of glucose concentration in our blood. And when it's chronic, when it's long-term elevated levels, that can actually lead to a variety of pathologies that are oftentimes characterized among diabetic patients. Among them is going to be failure or dysfunction of key organ systems, and this is going to be oftentimes characterized, excuse me, down at the microvasculature at the capillary level. So, for one example is there's diabetic retinopathy. What that means is that diabetic patients, there's a dysfunction because of the dysfunction to the capillaries within the retina. There's going to be blindness associated with some diabetic patients because their retina slowly becomes destroyed. In addition to that, there's diabetic nephritis. A key function is this filtration within the kidneys, where the capillaries are really important there. And eventually what ends up happening is that there's this elevated blood sugar level almost clogs up the nephrons and can create a lot of really inflammatory phenotypes within the kidney among diabetic patients. In addition to that, there's diabetic neuropathy. There's going to be a lot of nerve damage associated with diabetic patients. There's going to be loss of feeling within the extremities, and especially in really severe cases of diabetes, some patients, when they're bedridden, can't feel pain in some areas, and so they'll develop really problematic sores and ulcers because of that. And there's going to be a lot of associated cardiovascular issues among diabetic patients as well. Some of the key symptoms are going to be really related to one another. So polyuria is excessive urination. Polydipsia is constant thirst. Part of the reason is because this elevated sugar concentration in the bloodstream, when it gets to the kidneys, and the sugar is then filtered into the urine, it's going to create this osmotic pressure that's going to drive a lot more water to be excreted into the urine. And so as a result of that excessive loss of water due to this osmotic pressure, that polyuria is gonna drive polydipsia. It's gonna cause a lot of excessive thirst among these diabetic patients. What's interesting to note, going back to the previous slide, the term we use to characterize the class of all, the overarching term for diabetes is diabetes mellitus. Mellitus is a Latin term for sweet. The reason that's the case, for those of you who are pre-med, this might be, you might be thankful that you're pre-med now as opposed to a long time ago. The reason why it's called mellitus, because sweet is in there, is before the way in which patients were diagnosed with diabetes is that, again, sugar is being sent into the urine, and doctors used to diagnose them by smelling and tasting the urine to see if they're in fact diabetic. So thankfully we have blood glucose monitors that can substitute for that. There's also excessive weight loss associated with this because their metabolism and the energetics associated with all of their cellular functions is dysfunctional. That's going to create a lot of weight loss. It's going to affect growth, particularly among those who are first one class of diabetes, typically diagnosed between age 8 to 12. So it's going to affect their growth. And in addition to that, there's going to be a change in the cellular metabolism for like what key substrate is going to be used. Instead of now trying to use glucose that can't be taken up into the cells, lipids are going to become a key energy source here. And as lipids are metabolized to try and be used as to try and create ATP through lipid metabolism, it's going to create these ketone bodies or these ketones, and it'll put patients in a state of ketoacidosis, these excessive presence of ketones, and it actually creates a fruity smell in diabetic patients' breath. There are a few key complications that can emerge. We talked about retinopathy, the destruction of the retina. We talked about neuropathies, the nerve damage can be associated with diabetes. Throughout there's the cardiovascular component that we talked about really briefly. There's also a cerebrovascular component. As you can imagine, there's a lot of small capillaries that are present within our brain, and the ability for those capillaries to function properly is affected even within the brain. Hypertension, high blood pressure, there's going to be affected lipoprotein metabolism, again, because lipids are being used as an energy source here, as a key energy source. And in addition to that, there's going to be instances of periodontal disease, gum disease here. So what ends up happening is that there's going to be a class of bacteria that really thrive in these high sugar environments. And so that class of bacteria is going to actually prompt and promote gum disease. As you can imagine, I imagine a lot of us know people who are diabetic, and so there's this was back in 2017. I couldn't find any updated numbers, but as of 2017, the total costs associated with all of diabetes is 327 billion. I imagine by 2023, this is approaching probably 500 billion. For direct costs, back in 2017, it was 237 billion, and this is all just in the US. Which means if we go global, which over time, there's been higher incidence rates across different countries, particularly those in the Western Hemisphere, of higher rates of diabetes. Within the US, incidence is about 1.9 million per year, again, just in the US alone. And what's interesting is that there are some differences in the prevalence rates across different populations within just the US. Those originating from either African or Asian ancestry have typically lower rates of diabetes. Some of that might be attributed to differences in diet, but that's only one component that could possibly explain it. Within the US alone, there are varying rates across white, African American, and Hispanic populations, but it ranges from 7 to 14%. Interestingly, in some populations, particularly the Pima and Papago Native American tribes, rates can be as high as 50%. And that's particularly because of potential, an actual genetic predisposition that emerges in those populations. And so there's both cultural, there's both external and genetic forces at play that can drive the emergence of diabetes. Before we move forward, are there any questions here? So no one has questions about tasting urine? That just like, you guys are just going through that? Okay. So there are three types of diabetes. The first is type 1. This is an autoimmune destruction of the insulin-producing cells, the pancreatic beta cells. So there's an autoantigen on those specific cells. There's oftentimes proteins on the surfaces of different cells that are unique to them. That's one way in a research setting we can identify different cells through different experimental approaches. So there's a protein, there's something on the surface, forgive me, there's an autoantigen on these pancreatic beta cells that leads to their destruction. And so in type 1 diabetes, it's an autoimmune disorder that leads to the loss of these insulin-producing cells. So there's a lack of insulin being produced in type 1 diabetes. Type 2 is a much more prevalent category. The way in which it can be best characterized is that it results either from poor or defective insulin production or defective insulin signaling. One key phrase you'll oftentimes hear is insulin resistance. And so there's this one, type 2 diabetes can emerge from a few different ways. It's most commonly thought of as being as a result of being associated with obesity and metabolic disorders, metabolic syndrome. One thing that's also important to note is that it's also associated with aging. There are a lot of people who, as they approach their 70s and 80s, will actually become type 2 diabetic, merely because as they age, touching back on something we talked about in the previous class, touching back on senescence, the sheer fact that their pancreatic beta cells become dysfunctional over time. And so you'll see oftentimes a high rate of type 2 diabetes among older patients. Another example of type 2 diabetes is mature onset diabetes of the young. Previously, these were type 1 and type 2 were talked about with respect to when they were typically diagnosed. Some of you may have heard of juvenile diabetes. Type 1 diabetes was historically diagnosed in patients between 8 to 12 years old, but it's become more and more clear that some patients are diagnosed in their late teens. I've had a few friends that themselves were diagnosed first year of college. And so it's historically 8 to 12 years old, but can also be diagnosed in late teens. Type 2 diabetes is typically associated with adult onset diabetes. These are more antiquated terms, but it's more helpful to keep it with type 1 versus type 2. Not on this slide is also, what I forgot to say is the MODY, mature onset diabetes of the young, is a transcriptional defect that's inherited, a mutation that's inherited. And so that's the Native American tribes where they have rates that are as high as 50%. This is an inherited mutation that's passed down through that people group. One that's not listed here is type 3 gestational diabetes. That is diabetes that emerges in pregnant women. Typically, what ends up happening is that there's oftentimes really drastic hormonal changes that can affect insulin responsiveness among those patients. And what can actually be interesting, what can be unfortunate is that if there are multiple bouts of gestational diabetes with subsequent pregnancies, it can actually lead to type 2 diabetes in some cases. All right, before I move on, are there any questions here on the different types of diabetes we talked about? Mm-hmm. Yeah, the question is, in cases of type 3 diabetes, can it be passed on to the fetus? Typically, what ends up happening is that there are as long as it is managed properly, again, like we talked about how all of these symptoms, not symptoms, all of the complications that emerge from diabetes involved with effects like through the capillaries, through the vasculature, and the placenta is a highly vascularized organ system. Yeah. And so it can be there, it can emerge, there can be a lot of complications that emerge just during pregnancy if it's not addressed properly. That can lead to death of the mother or the fetus. And so as long as it's properly managed, there's not going to be any predisposition in the fetus to develop diabetes in the future. Are there any other questions? All right, let's go ahead and jump into transcription. So we're first going to look at transcription at a high level, and then we'll go ahead and jump into looking at the different stages of transcription. Give me 1 second. So what we have here in the top portion here is the different segments that make up what is where transcription is taking place. We have one single transcriptional unit, which in eukaryotes is typically one single gene, and prokaryotes can actually be multiple genes, but are regulated together as a result of being transcribed together. Transcription begins at the promoter, which is highlighted to the left of this transcription unit, and it ends at the terminator sequence to the right of the transcription unit. So what ends up happening here is that the RNA polymerase will bind to the promoter region, and it'll unwind the DNA there. So what's important to note is that this is very different from replication. We talked about all of the things that DNA polymerase is unable to do on its own, all the things that it needs help with. It can't unwind, it can't initiate. What's important to note here is that there are no helicases, there are no gyrases, there's no supercoiling. The unwinding is a lot smaller than what happens in replication. So as kind of an analogy to help illustrate this, you can envision it where if you lose something in the couch, just sticking your hand in through the cushions and like running your hand through, that would be transcription. Replication would be taking the cushions all the way out and basically unwinding the couch, for lack of a better phrase, taking out the cushion. So with transcription, we're basically, it's similar to like putting your hand in the couch and just running it through. RNA polymerase is just going to the promoter, opening up a little bit to access the nucleotides, and it's going to run across during the course of transcription. And so what's also important to note is that we're going to have our NTPs, our nucleoside triphosphates, that are the building blocks for the mRNA strand that's going to be synthesized during transcription. And the way in which it's going to be synthesized is that there's going to be, when the DNA is unwound, we have two strands here. One strand is going to be the template strand. Similar to replication, there's going to be one strand from which a new strand will be synthesized. The difference here is that it's going to be an RNA strand, an mRNA strand, that is complementary and anti-parallel to the template strand. And it synthesizes the mRNA in the 5' to 3' direction, which means it reads the template strand in the 3' to 5'. What's also important to note is that since there's another strand of DNA above, what's important to note is that is going to be called the coding strand. And it's called that because it'll match the mRNA strand that's synthesized, because that other strand is going to be also anti-parallel and complementary to the template strand. The main difference is that the coding strand will have thymine, T's, and the mRNA strand will have uracils or U's. And so again, just to repeat, the mRNA strand is synthesized in the 5' to 3' direction. It'll be anti-parallel and complementary to the template strand, and it'll almost nearly identically match the coding strand. The mRNA strand that's synthesized during transcription will nearly identically match the coding strand, with the difference of being, instead of thymines, you'll have uracils in the mRNA strand. And so over the course of transcription, we'll have elongation, the growth of that mRNA strand that's growing in the 5' to 3' direction, as more NTPs and nucleoside triphosphates, or the building blocks, are used and added into the growing mRNA strand. And then once it reaches the terminator site, there's an actual cue there that prompts transcription to end, for RNA polymerase 2 to stop synthesizing the mRNA strand, and for now this mRNA transcript to be cleaved and released, concluding transcription. And so we're going to take a moment to look at some of the different steps of this entire process to get a little bit more detail. So we talked about the promoter. Oh, yes.
Are we good to like?
Yeah, so the question is it okay for us to equate a transcriptional unit with a gene? That is okay in eukaryotes, and that's true in eukaryotes. One transcriptional unit equals one gene. In prokaryotes, that's not the case. In next class, you'll actually see a few instances of how one transcriptional unit in prokaryotes will encode for multiple genes, because they'll all be regulated together and they're all going to be affecting one another in the regulation of transcription. But to to provide a simple answer to your question, in eukaryotes, one transcriptional unit equals one gene. In prokaryotes, that's not the case. So we talked about promoters being the site where transcription begins, where RNA polymerase will bind and initiate transcription. What's important to note is that there's going to be, excuse me, there's going to be a few key distinctions between eukaryotic and prokaryotic transcription, one of which is what we just talked about, the differences between the transcriptional units. And just to repeat again, the template strand is what's going to be used as the guide during transcription, as the template here. Whenever we talk about promoters, it's actually upstream, or usually just above or away from where transcription originally begins. As you can see here, we have something that's called the start point right here, where it has this +1 symbol. That is the first nucleotide at which transcription occurs. Upstream of that on the coding strand, or to the left, or in the 5' direction, that is the actual promoter. So the promoter is not part of what's going to be encoded in the subsequent mRNA transcript, but rather it's the site at which RNA polymerase is promoted to bind to then promote subsequent transcription of the transcriptional unit downstream from the promoter. And what we've learned about the promoter is that it's actually, we've actually been able to identify and determine specific sequences that are conserved across species. And so we'd actually call it a consensus sequence. So we only talked about consensus sequences in the context of replication, or the context of origins of replication. What we can actually say now is that we can now take a step back and think more broadly of consensus sequences as being these conserved, bless you, these conserved and known sequences of DNA that provide a signal to elicit some process. And so in the case of transcription, there are regions of the promoter that are well characterized where we know these conserved sequences. For example, one of them within the promoter, and we'll be talking about this within the context of the coding strand here, one of which is called the Tata box. And so that's a part of the promoter that is a key part of a key and conserved sequence across transcription. So if we look, oh, yes. You said, what's the difference between the promoter and, you said, the primer?
Yes.
So the RNA primer, is that what you're talking about?
Yes.
Yeah, so the RNA primer is key for the ability for RNA polymerase to initiate its polymerase function for replication. The promoter is key. And so what I should say is the primer is key for promoting the polymerase activity. This promoter is key for being the site at which RNA polymerase binds to the DNA so that transcription can begin. Does that help clarify? Okay. So again, the conserved sequence, the consensus sequences within the promoter have been identified bioinformatically, where we can look at the different promoters across different species and find and identify what has been conserved across species so that we can identify and see that there's these specific sequences, among which is the Tata box within the coding strand, that helps us understand what is a key component of our promoters. So we'll start by looking at transcription at the initiation step. Okay, so with initiation, a key thing to note is that our RNA polymerase will bind to the promoter, and at the starting point, that's the point at which of our DNA where it will be the template strand will be used to synthesize the anti-parallel and complementary RNA. It's going to begin laying down and creating, not laying down, it'll begin creating the new mRNA strain that's complementary there. But the RNA polymerase is going to stay put. It's not going to start moving yet. As it starts synthesizing these base pairs, approximately 9 of them, once they've been added, then elongation can occur where now RNA polymerase will start moving across our DNA. And so what's key to note is that it's using nucleoside triphosphates, NTPs, as the building blocks for the mRNA transcript. And that it also, for initiation, it's key to note that the RNA polymerase stays put as the first pieces of the mRNA transcript is synthesized. Once enough of it has been synthesized there, then RNA polymerase will start moving along the DNA. And again, it's worth noting, there are no helicases, there are no gyrases, and there's no supercoiling occurring during transcription. During elongation, as the mRNA transcript is being further synthesized during this process, we're going to have RNA polymerase moving down the DNA and unwinding it. Again, you can envision if you're looking for something in a lost couch, you put your hand in between the cushions and slide it across. It's the same thing. RNA polymerase is sliding in, wedging open the DNA a little bit, and sliding across, like unzipping it a little bit by a certain amount as it slides across our DNA. And it continues to add nucleoside triphosphates, approximately 12 at a time, as it moves along. And what's also important to note is that as it moves along, the DNA behind it is being reannealed. It's being wound back up and binding back together. And that it's important to note that this RNA that's being synthesized is happening in the 5' to 3' direction. And another thing that's worth repeating again is that because it's complementary and anti-parallel to the template strand, our RNA will be matching the coding strand, the other strand here. But again, the only difference is that instead of thymines, we're going to have uracils on our RNA here. So to terminate all of this, once our RNA polymerase gets to the terminator site, what ends up happening is that there is encoded within that terminator site this self-complementary sequences by which because they're self-complementary and because oftentimes it's energetically favorable for nucleotides to anneal together if they are complementary, this self-complementary sequence will then cause, for example, if we have CCCCC, another sequence of RNA that's synthesized, and then GGGGG, they will bind, it'll loop around and bind upon itself, creating this hairpin structure within the mRNA that was just synthesized. This hairpin structure is a secondary structure that's a key cue or a biophysical signal to RNA polymerase that transcription needs to end here. And so once this self-complementary hairpin structure is formed, that is the key signal for termination to end, and RNA polymerase will then leave the DNA, and that'll be the end of transcription there. As for RNA polymerase, we actually have 3 species of RNA polymerase, and because we have 3 species of RNA, you can envision how it matches up. We have one specific species that makes our RNA, the ribosomal RNA, that's RNA polymerase 1. For our mRNA, RNA polymerase 2 is the key actor there. And so RNA polymerase 2 is a key one for you to know because that's when it's going to be critical for you to remember for transcription. And finally, for transfer RNA, the key one there is RNA polymerase 3 or RNA pol 3. And so what's important to note is that each of them will end up using a different a different promoter structure because of their because each promoter will recruit a different species, will be inclined to recruit a different species of RNA polymerase. So the key one for us to focus on today is RNA pol2, RNA polymerase 2. And we're going to talk about the promoter, the properties of the promoter that specifically recruits RNA pol2. Yes. Yeah. So what self-complementary means is that there are sequences within that one strain of mRNA where they'll actually be complementary to each other. There's going to be there's going to be the ability for it to bind back upon itself because there's a stretch of, so for simplicity's sake, there'll be a stretch of, for example, a stretch of C's, the cytosines, and then a stretch of guanines, which are complementary. C's binds to G's and A's bind to T's. Or in the case of mRNA, adenosine binds to uracil, A to U. And so in this hypothetical example, if we have an mRNA strand where the terminator sequence encodes for a stretch of just cytosines, stretch of just C's, and then say a couple dozen nucleotides down, there's a long stretch of G's, that will be complementary to one another and promote the looping and binding of those complementary sequences to then bind together and create this hairpin structure. So the hairpin structure is mediated or enabled because of these stretches of mRNA that are complementary to one another, C to G, A to U. So, so the properties of the promoter that recruits RNA pol2, we have what's called the initiator sequence. It's this stretch of DNA that surrounds the start point. Again, the start point is denoted by this little black box that has +1 above it. That is our first nucleotide that's going to be our first DNA nucleotide that's going to be used actually during transcription. And around it are these stretch of pyrimidine nucleotides. That's going to be cytosines or thymine. And so you'll have a couple to the left of it, and then you'll have another 5 to the right of the start point. And so these nucleotides that surround the start point, we call that the initiator sequence. In addition to that, we're going to have the ta-ta box to the left of the initiator point. And so that's going to be about 25 base pairs upstream or towards the 5' end. That's going to be, and these two together, the ta-ta box and the initiator sequence together make up what's called the core promoter. That's going to be key because that's going to be, that's a consensus sequence that's conserved across species that's well defined. There's going to be different components that have been that are depicted here. We have the BRE, the B recognition elements. We have the DPE downstream. We have which are the downstream promoter elements, and those have a little more flexibility across different species. But the core promoter, the ta-ta box initiator sequence, are well defined, conserved, and serve as a consensus sequence here that is important for the promoter in transcription. And so there's an entire class of proteins that are actually able to move into the nucleus and bind DNA and promote transcription. They're actually called transcription factors. And they're really key because they bind to the ta-ta box and actually can help promote more transcription. And so I'm sure a lot of you come across different proteins in your projects that are transcription factors, or some of you work in labs where there's a favorite transcription factor that the lab perhaps studies or manipulates. And so a transcription factor, again, is a key protein that's able to actually bind to DNA at the ta-ta box before RNA pol2 is even there, and it's actually able to promote more RNA pol2 to more easily and readily bind to the promoter. What's key to note is that a lot of those transcription factors are typically activated. There's a phosphorylation event that triggers them to go into the nucleus and bind to the DNA. There's a subclass of transcription factors that actually do not require activation, but rather if the but it's an important class of transcription factors that are required in order for RNA Pol II to bind to the DNA. And those are called basal transcription factors. For RNA Pol II, that transcription factor, that basal transcription factor is called TF2D. And so it also binds to the Tata box. But what's important to note is that if the Tata box is available on a specific gene, it will readily bind. And so we talked about how our DNA is compacted in certain structures within our chromosomes. And what that means is that, what that means is that certain regions of our DNA are going to be readily either available, accessible, or it's going to be compressed and compacted and not accessible. And so there's going to be, for example, there's going to be points, whether it's an immune cell secreting inflammatory proteins or cytokines, and the way in which that happens when these immune cells are activated, that region of the DNA where there's all these signaling events that leads to our DNA being either compressed or moved and more readily available. If that region of the DNA is available for a certain gene, TF2D will bind to the Tata box there. And so the key thing to note is that if the core promoter is available, TF2D will bind to that core promoter there. And so typically what will end up happening is that once TF2D binds, if there are other transcription factors that are not basal, but rather can be activated, if they are activated, there's going to be the recruitment of more transcription factors to the core promoter site. And again, the more transcription factors that bind to a core promoter, the more easy it is for RNA Pol II to bind. And so they're going to promote transcription there. And so once these transcription factors bind, and they always do so before RNA Pol II. So once they bind, they're going to promote the recruitment of RNA Pol II there to the promoter, which will then promote subsequent transcription across that gene, across that transcriptional unit. I'm going to stop here and wait for a couple of questions. Do we have any questions here? Yes. No, they're not only in eukaryotes, but the main eukaryotic basal transcription factor that is well characterized as TF2D. I'll wait for another couple questions because I think there was a lot of content we just went over. Yes. Mm-hmm.
What was
Yeah, I think I went over this a little too quickly. Thanks for asking that. So the PY2 means, so I'll take a further step back. Our nucleotides in our DNA are categorized into two groups. We have purines, which are guanine and adenosine, and pyrimidines, which are cytosine and thymine. The PY stands for pyrimidines. So that is going to be that's going to be either cytosine or thymine. And so PY2 just means two of them, two pyrimidines. And then to the right, where you see PY5, that just means 5 pyrimidines. So either some combination of either cytosine or thymine. Thanks for asking that. Yes. Great question. So the question is the consensus sequences that we've talked about so far in this class, as related to replication, was only in prokaryotes. Is what we're talking about in transcription relating to only prokaryotes or prokaryotes and eukaryotes? In the context of transcription, it's both. So eukaryotes also have these consensus sequences that have been well defined at the promoter, primarily the core promoter, that constitute the Tata box and the initiator sequence around the start point that are really well defined. And so again, in the context of replication, we had a very specific definition of consensus sequence. We talked about it within the context of being at the origin of replication. Now we're going to take a step back and we're going to have a bigger definition of consensus sequences being these conserved sequences of DNA across species that that serve as a signal for some biological process to occur. And so here, these consensus sequences serve as a signal to recruit both transcription factors to bind as well as RNA Pol2. All right, if there's, I'm going to press on, but again, if anything doesn't make sense, feel free to shoot your hand in the air. So at this point now, we have a fully synthesized mRNA strand. What's important to note is that it's a slightly immature mRNA strand, and we'd actually call it, the name we have for it is called pre-mRNA. What's important to note is that there are important modifications that are needed to ensure that the mRNA strain is not degraded. If any of you, whether you're in Ideas Lab or if you work in a research lab, and if you've done qPCR or if you've handled mRNA at all, you can appreciate that it is really easy to degrade. It's really finicky. And so there are modifications that have been derived over evolution to help ensure that our mRNA stays stable and is not easily degraded. The three key modifications here is a 5' cap, a poly A tail, and we've already alluded to this a little bit in the previous lecture, but also splicing events as well. The first modification is a 5' cap. It's a 5' methylated cap. What it is this 7-methylguanosine that's added at the 5' end. And what's interesting about it is that it's a backwards 5' cap. What that means, the reason why it's backwards is because when we talk about 5' to 3', that's all with respect to how each nucleotide is connected across the carbon ring from each adjacent nucleotide. So it's bound one 5' carbon is bound to the 3' carbon of the next adjacent nucleotide. Here, with this 5' cap, it's bound to the 5' carbon here, and then bound to the 5' carbon of the next adjacent nucleotide. So it's a backwards 5' to 5' cap, and it's added during elongation. So it's not at the end when transcription is done, but it's during the process of transcription. What's important to note is that this 5' cap serves a few key functions. It is a signal for nuclear export. So once this mRNA strain has been synthesized, it needs to be transported outside the nucleus. The nucleus is where transcription is occurring. It needs to be moved and shuttled outside the nucleus so that it can be used for translation. This cap will help provide protection against degradation. And there's going to be a slide that kind of helps illustrate why in a second. And again, another important thing is that this is actually an important binding site for ribosomes, which is really critical for translation. There's actually a capping enzyme that mediates the addition of the 5' cap, and it's only associated with RNA Pol2. And the reason why this is important is because we only want our mRNA to be associated with these functions that are attributed to the 5' cap and not necessarily to our other species of RNA. In addition, we have our poly A tail that's added. Another term for this is polyadenylation. Many adenyls that are added. So one thing that's important to note is that our transcripts that are made during the course of transcription are thousands of base pairs long, tens of thousands of base pairs long. And so once we hit the terminator sequence, and as this hairpin loop is forming, until that hairpin loop is actually created and for RNA Pol2 to process that biophysical signal, RNA Pol2 keeps moving and synthesizing and elongating this mRNA strand. So there's going to be a fair amount of nucleotides that are at the end, at the 3' end, that are going to are creating a transcript that's a little bit too long. And just for clarity of language, I think I've made not made a mistake. I keep using a term here that I want to clarify. When I say transcript, I'm just talking about the mRNA strain that's synthesized during transcription. So if you ever hear transcript, that just means the mRNA strain that's synthesized as a result of transcription. So what ends up happening is that there's going to be a key enzyme that mediates a couple key functions here. It's called a poly-A polymerase, or PAP. And so it's going to find a specific signal, a specific consensus sequence. And here that's going to be AAU, AAA here, close to the 3' end. And that will serve as a signal for PAP to mediate its function. So the first thing it's going to do is that it's going to cleave the transcript about 10 to 30 nucleotides away from that consensus sequence. And once it clips it there, then what's going to end up happening is that PAP is going to add a poly-A tail or lots of adenosines there to the end. What this poly-A tail does is that it does two important things. It helps protect the transcript again from degradation. That's going to be the overarching theme for a lot of these modifications. We want to protect the mRNA because it's typically encoding something important. So it helps protect it from degradation, and it's really key for translation. The one distinction between prokaryotes and eukaryotes here is just the length of the poly-A tail. It's typically about 20 to 30 nucleotides long in prokaryotes and about 20 to 100 nucleotides long in eukaryotes here. And another common question we've gotten here is why adenosine, why A? An important thing to note is that the base for adenosine is the same base for what makes up ATP or ADP or AMP. And so that's a molecule that is readily available throughout our cells. And so because it's abundantly available, it's commonly, it is what that's I think like an evolutionary advantage of it being what's being added to the end of our mRNA. So to help put all of these modifications in context of how they help protect the mRNA strand, the transcript from degradation, we talked about exonucleases and endonucleases in the last two lectures. There is a class of nucleases that are called exoribonucleases. They specifically target RNA-based nucleotide strands, RNA strands. And because it's an exoribonuclease, it focuses at the end. It'll basically eat away at the end. And so that's why we have these two modifications at the ends of our mRNA molecule, mRNA strand here, our 5' cap and our poly-A tail. Exoribonucleases are actually really important because a lot of viruses are RNA based, and so it's a natural mechanism to help protect against that class of viruses. But here we needed to make our biologically it was needed to make sure that those protective class of enzymes are not targeting our own RNA. And so the 5' cap and the poly-A tail helps protect against that. And so what we have here are these poly-A binding proteins, this class of proteins that help bind to the poly-A tail and help protect it on that end. And we have our decapping protein that'll help remove the cap to enable translation. And so what's also important to note is that these mRNA strands don't have a super long half-life. They don't last forever. And so once they're synthesized, they need to be used for translation. Otherwise, there's going to be a point at which they're going to be degraded. And so as an example here, we have slowly the whittling away of the poly-A tail and the loss of these poly-A binding proteins. And you can think of the loss of these components as almost a timer. So the decapping protein kicks off the 5' cap, and that enables exoribonuclease to mediate the degradation of the mRNA. And so once an mRNA strain is synthesized, there's almost a timer that's set off for it needing to be used for translation, or otherwise it'll undergo degradation during via natural processes here. I'm going to stop here and wait for a couple of questions because, again, I think we went over a few key concepts, but I want to leave space for a couple of questions. Yeah, go ahead.
Uh,
Yeah, so let me go back to the beginning of this of the illustration. So the poly A binding proteins, their main function is to bind to the poly A tail and help stabilize that end of the mRNA transcript. We have the 5' cap that helps protect the 5' end. We need something that protects the 3' end. So the presence of the poly A tail is a signal for these poly A binding proteins to bind to that 3' end and protect that end. Exoribonucleases want to work from the ends. So the 5' cap protects the 5' end. The poly A tail, with the presence of these poly A binding proteins, protects that other end. Did you also ask about the decapping protein? And so that helps. The decapping protein is really important for mediating. It's really important for translation. And so once that decapping protein kicks off the 5' cap, this mRNA transcript is vulnerable, for lack of a better phrase. And so it needs to be used for translation. Otherwise it'll undergo degradation. It'll undergo degradation fairly quickly if it's not used. Yes. Yeah, so the question is, when is the 5' cap added? So let me go back to the elongation slide. So it's added during, sorry, I'm gonna go back a little bit. So it's added during elongation. The 5' cap is added during elongation. So we have, as RNA polymerase is traversing across our DNA, transcribing this gene and creating the mRNA, there's going to be a piece of the mRNA that is, for lack of a better phrase, hanging outside of RNA polymerase 2 as it's being elongated and synthesized and adding on more nucleotides. That piece that's hanging off is the 5' end. So it's growing in the 5', it's creating the mRNA in the 5' to 3' direction. So that 5' end is available. And so during elongation, the capping enzyme will add the 5' cap to the 5' end during elongation. I appreciate you asking that because I think I kind of went over that pretty quickly. So I appreciate that. All right, we're going to go ahead and press on. So up to this point, we've talked about, and recap a couple times, but it's worth saying it again. We've talked about protein domains versus protein subunits. Protein domains are parts or regions of a single polypeptide protein where there's going to be different functions of that single protein. And so, for example, we talked about with enzymes that there's going to be, an allosteric domain or a catalytic domain. They're part of the same single stretch of amino acids, single polypeptide protein, whereas subunits are these different individual proteins that come together to form a macromolecular structure. For example, hemoglobin, there's two dimers that come together to form this large macromolecular structure. And so each subunit is going to be encoded by a different gene. Our domains of a protein are encoded by different exons typically. It could be, sometimes it could be a single exon that encodes for a domain or a combination of exons that encode for a single domain. But as we talked about in the last lecture, that we have both exons and introns within a single gene, and that during splicing, we have the introns that are spliced out. And so each of these exons here typically represent a specific domain within a single protein chain or single polypeptide. And again, it can be that a combination of exons, it could be that exon 1 and 2 constitute one domain, but a lot of times it's typically one exon to one domain. So it's during splicing whereby these introns are typically removed from the primary transcript. So our primary transcript or the pre-mRNA has the introns present within the mRNA, or I should say, it has mRNA that is complementary and anti-parallel to the introns of the template strand. What's important to note is that typically the when we in bioinformatics, because we've mapped the human genome, we've numbered every single nucleotide that corresponds to each gene. When you look, and because of that, we can also know the nucleotides that correspond to the mRNA. So when we look at the primary transcript or the pre-mRNA versus the more mature mRNA, we actually don't count or provide numbers to the intron associated regions of the mRNA. And so you'll see that it's numbered from 1 to 46, but it jumps from 30 to 31 within the first intron and jumps across and skips that intron as well. And so when it's been appropriately spliced and all of the exons have been merged together, we have a single contiguous mRNA transcript that is only corresponding to the exons of the template strand. What's important to note is that there's a lot that's known about this process of splicing, and there's a lot that's unknown. What's known is that there are specific sequences that are serve as targets for where splicing occurs. And so we typically call these conserved sequences the splice sites, and they're typically where at the beginning and end of the introns. And so there's usually a 2-base sequence on the 5' end of the intron and a 2-base sequence at the 3' end. And so the 5' end is GU and the 3' end is AG. And typically they are conserved across all introns here. And you can, and the reason by which that's important to note is that this process of splicing is really important, and it's important that it's consistent so that there aren't variations, and there aren't, what's a good way to say this? Because splicing is so important, and because a lot of times proteins that undergo, there's a large number of proteins that, if they are spliced incorrectly, can lead to dysfunctional truncated forms of the protein that can lead to disease. This needs to be fairly conserved. And so usually the introns, these conserved sequences at the 5' and 3' end are fairly conserved here. And so these 2-base sequences cause the formation of the structure called the spliceosome. That's a combination of different proteins and RNAs. The structure of proteins and RNAs together, we call them small nuclear ribonucleic particles, or SNRPs. And so you might hear them called SNRP1, SNRP2, and such. And so what's important again to note is that we know the sites, we know the splice sites well. We don't know a lot of the regulation that leads to different splice variants. And so to at least focus on what is well known and what's been well characterized, focusing on moving from starting at the top left here, and we'll move across. I'll take a moment to say really quickly, I am not asking you to study and memorize every single piece of this process, but rather appreciate the big picture, understand what perhaps characterizes the splice sites. And yeah, so what I'll say here is that at the 5' end of this one intron here, we have the first SNRP, U1, that binds to the GU site. We have the next SNRP, U2, that binds not at the AG down at the 3' end, but upstream at what's called the branch point sequence, this adenosine right here, and you'll see why momentarily. Once those first 2 SNRPs bind to the mRNA transcript, that'll actually promote the recruitment of more SNRPs, U4, U5, and U6, to begin forming the spliceosome here. So these, this collection of proteins here actually constitute what we call the spliceosome. Once they're all together, for lack of a better phrase, once the gang's all there, what ends up happening is that UG here, or sorry, GU, will actually be caused to loop around and bind to the branch point sequence. The reason why that's important is because it'll actually enable the pinching off and removing of the intron. And this whole structure here, once the 5' end binds to the branch, is bound to the branch point sequence. We call this the lariat structure. The intron, the shape it's in, is called the lariat structure. And so once that happens, then what we have, what I should say is once the branch point sequence, or once the lariat structure is formed, that happens by when the 5' end is clipped away from the exon. Once the structure is here, now the spliceosome will cut the 3' end of the exon, of the intron, forgive me, and wedge together the two exons and excise that intron. Once it's been excised, it's a collection of nucleotides. It's an mRNA of just the intron that is vulnerable to exoribonucleases, so it'll undergo degradation, and all of those nucleotides will then be recycled for other transcriptional events. Again, the important thing to note here is that we have the binding of SNRPs at the splice site at the branch point sequence that causes the spliceosome to then be recruited and formed, forming this structure where the GU binds to the branch point sequence. And one thing I should note is that if you look at the bottom left, the GU are the first two nucleotides of the intron, but it looks like there's this stretch of almost what looks like more nucleotides there. That's a bit of a illustration error. The GU, there are no nucleotides in the intron to the that are upstream from those two sequences there. And so the key thing to note is that it gets pinched off, forms this structure, and excises the introns here. I'll stop for a couple of questions here. Yeah.
With the sequences, like the two big sequences that where
Mhm.
are like G and AG like really common? Or like are they directly together? And like if not, how do like the nerds differentiate the one you?
Yeah, that's a really good question. And the whole process of splicing is an entire area of research. As someone who's not in that area of research, I think your question is part of the reason of how we get different splice variants. That there are AGs and GUs that might be present or likely present within the pre-mRNA, this primary transcript, that can cause, perhaps in this form, this exon will be excised out and you get this variation, the splice variant. My hunch is that that's how we get different splice variants. As someone who is not in splicing research, that's the best answer I can give you. That I think what you just asked, like if we have these splice sites 2-base sequences in the actual exons or elsewhere within the within the mRNA transcript, what can that cause? I think that's how we get different splice variants. But that's the best answer I can give to your question. Are there any other questions? Yes.
Like, you explain what you meant in slide.
Yeah, so when I say they're 100% conserved, what that means is that across all introns, they are going to have at the 5' end a GU sequence and at the 3' end an AG sequence. And I think to perhaps maybe add on to the previous question, if there is likely a GU sequence, but not a specific branch point sequence, or if there's a GU on the 5' end, but not an AG on further down, if we're like sitting in an exon where there's likely going to be an AG, like that's statistically likely. But there might not be an AG nearby whereby the spliceosome can actually promote splicing. So that'll not promote splicing to occur there. But because the process of splicing is so important, it's evolutionarily conserved across species that these two sequences constitute nearly every intron within mRNA. Does that help clarify? Great. Okay, with that, I think I've kind of held you guys 2 minutes long the last few times, so we'll break 5 minutes early. But if you have any questions, I'll be hanging back. And thanks for your time.